Hi all. Today we are going to discuss about chapter number 5 which of 9th standard, that is fundamental unit of life. That means you have studied about this. That is it's this here we are going to discuss about in detail one by one about what is cell, who discovered cell, what are the functions of cell, what are different types of cell, those things. So first we will see what do you mean by a cell. There are some stories behind this. We are that means historical part. We are not entering into those things. So first we will see what is a cell. Cell is a structural and functional unit of life. That means the cell gives structure and it performs a function. Because of that it become a unit of life. That means if there is no cell, there is no tissue. If there is no tissue, there is no organ. If there is no organ, there is no organism. And if there is no organism, there is no life. So cell performs that basic thing. And according to the historical point of view, the first living cell was discovered by Antony von Leeuwenhoek. Before that, there was no evidence about any type of microscopic thing. And the first non-living cell was discovered by Robert Hooke. And he named it as cell. And that word cell is a Latin word. If you are translating it into English, it means compartment. Compartment means very congested, like this, very congested. And when he observed under the microscope, he has seen it as a dead one. That means non-living. But Antony van Leeuwenhoek was seen as living one. But Antony van Leeuwenhoek was not knowing what is that, so he forwarded his writings to Robert Hooke, who was the professor or a scientist in Academy of Sciences in England. Then later, whatever the Antony van Leeuwenhoek was found, Robert Hooke has confirmed it, and he called the term as cell, that is a compartment, which means a compartment. And when we are coming to the basic of cell, there are three theories are there which was put forward by three different persons. One is Matthias Leiden, Schwann, and Virchow. What they found, what they said is, after discovering all this, that the cell means the cell performs a structural functional unit of life and everything, these three perform a basic cell theory which is called as cell theory. That means all the cell they follow this. What they performed is, first theory says that cell is the structural and functional unit of life. All organisms are made up of cell. These two theories was given by Schleiden and Schwann in those two different times. They were European scientists, they were given that. And Virchow, another person, added one more point to it, that is all cell arise from a pre-existing cell. That means cell division, reproduction, all the things comes inside that. So these are the things. That means any if we have to call it as a cell, all the cell, that means a particular cell has to undergo or follow these three theories. That is, cell is a structural and functional unit of life. All organisms are made up of cell. All cell arise from a pre-existing cell. These three theories they have to follow. The smallest cell, we'll discuss something about according to the size of the cell, which is the smallest one, which is the largest one, which is the longest one. The smallest living cell is mycoplasma. Still, we have something down also. And the largest cell is an ostrich egg. Living cell is an ostrich egg. And the longest cell is a nerve cell. And in our human body, the smallest cell is a sperm, and the largest cell is an ovum. And when it comes to the number of cells, when we are checking with the number of cells, the according in the type of organisms, if we are trying to divide it according to the number of cells, There are two parts are there. One is known as unicellular, another one is known as multicellular. Otherwise, unicellular we can say it as prokaryotic cell, and multicellular we can say it as eukaryotic cell. We have to study what is the difference between prokaryotic cell and eukaryotic cell, that we will deal. Next one, according to the shape and size of the cell. If you see a cell, there are some cells are there which is oval in shape. For example, egg. There are some cells are there, some they are spherical in shape, some are there in rectangular in shape, some are there spindle shape, some are irregular in shape. For example, amoeba, which are already studied, other things are present inside the tissue. So we have to get the names. We will study about that in later. So I will say one more thing. Some of the cells are oval, some of the cells are spherical, some of the cells are rectangular in shape, some of the cells are spindle in shape, some of the cells are irregular in shape. An example for an oval cell is an egg. An example for an irregular cell goes to the amoeba. So other things we will discuss. Now we are moving to a typical structure of a cell. I have drawn this diagram with a lot of limitations. So we have to go one by one as possible. This is a structure of a eukaryotic cell, and this is the structure of prokaryotic cell. Here I have drawn a eukaryotic animal cell, typically an animal cell. This is a eukaryotic, prokaryotic, that is a bacterial cell. So if you see inside this eukaryotic cell, plant cell also comes because of my limitations I have not drawn. So if you are seeing a eukaryotic cell, it is having a plasma membrane. That means these are some of the fundamental difference between a eukaryotic and a prokaryotic cell. And why we are calling it as eukaryotic cell and why we are calling it prokaryotic cell, because in eukaryotic cell, genetic material, what is present to the middle, is surrounded with a nuclear membrane. And if you are seeing in a prokaryotic cell, genetic material is naked. So pro means ancient, karyo means nucleus. So ancient nucleus cell, so they are prokaryotic cell. U means new, karyo or karyota means nucleus cell, which means a new cell. So if you see the basic difference between a prokaryotic and eukaryotic cell, a animal cell will be somewhere around 500 millimeters. That means 100 to 500 millimeters. And if you are seeing a prokaryotic cell, it will be somewhere around 0.5 to 2 millimeters. It depends upon organism to organism. And another difference is, if you are seeing an animal cell, which is a eukaryotic cell, they have membrane bound organelles. We have to study each and every membrane bound organelles. We will study that later. When you are seeing the membrane bound organelle, you can understand that organelles means little organ. That means they are the subunit inside a cell which is having some functions to do. They have some functions to do. And many of these membrane bound organelles are absent inside a prokaryotic cell. This is one of the major difference. That means after the nucleus, that means it is have a well covered nuclear envelope, and here the nucleus genetic material is naked. So it is, and if the genetic material is naked, we will call it as a nucleoid. If the genetic material is covered with the cell membrane, sorry, nuclear membrane, we will call it as nucleus. Then, and if you are coming to the cell organelles, many of the membrane bound cell organelles are absent in prokaryotic and it is present in eukaryotic cell. After that, if you come to the membrane bound organelles, endoplasmic reticulum is present. That means a structure that connects between the nucleus and the plasma membrane. That endoplasmic reticulum is present inside the animal cell, but it is absent inside the prokaryotic cell. And here they have mitochondria, which is called as the energy producing or powerhouse of a cell. Here it is absent. And lysosomes are present inside the animal cell, but instead of lysosome, few organisms have some other than they have presence, they have contractile vacuoles which controls the osmoregulation inside the cell. Apart from that, if you see an animal cell, they don't have plasmid and they have plasmid. Plasmid means extra cytoplasmic genetic material. That means apart from this, there is a circular structure is there, structural genetic material structure is there, which carries some other informations also apart from the uh, middle of the middle genetic material. Then, and if you are seeing another one more thing, they have here the ribosomes are attached to the endoplasmic reticulum, which are responsible for synthesizing the proteins. And here if you see, the ribosomes are scattered because they don't have endoplasmic reticulum. Whenever they want to synthesize protein, they will come together, they will come near to this genetic material and they will do the function. And if you see here, some of this prokaryotic cell, apart from the plasma membrane, they covers a capsule, that means an extra covering, which gives protection for this prokaryotic organism. So there is one more thing, if any organism which is having capsule, they are capable of virulence, that means they can cause disease. And this capsule is absent in animal cell because animal cells don't cause any diseases. And these capsules also help this bacteria to survive in a very harsh conditions. So if any organ, for example, for example, one of the best example is food poisoning. It is caused by a bacteria, and in our stomach we are having HCl. But these bacteria are not killed by your HCl. It is because those bacteria which are causing food poison, they are covered with these capsules. Then some bacteria are movable, they can move from one place to another. If they are moving, they have flagella. Apart from this, there's a small structure that arises from the capsule or the plasma membrane, which is called as the fimbriae. And on that fimbriae, if small thread-like structure again is present, like this, which is called as cilia. So these are some of the main differences between the difference between a prokaryotic cell and a eukaryotic cell. And in eukaryotic cell and prokaryotic cell, we can see some other important factors known as cytoskeletons. Cytoskeleton means, if you consider this as an animal cell, these animal cells, whenever they undergo pressure, whenever they relax, what will happen is it has to maintain its structure. To maintain its structure, their boundaries are connected with cytoskeletons. Cytoskeletons mean these skeletons, they don't allow the shell to shrink much and they don't allow the shell to expand more. If anything happen like that, the cell will burst off. That means this full structure will collapse. And when we are coming into the prokaryotic cell, they also have these type of cytoskeletons over the surrounding. Some of them they have, so they also do the same function. Apart from that, some of the prokaryotic cells are contractile vacuole. Contractile vacuoles are helping to do the osmoregulation. That means if any, since they are, most of them are living in water, in aquatic condition, there's a chance for the entry of water through this plasma membrane to the process of diffusion. So the contractile vacuole functions like a motor, and it will throw off the extra water that is coming from outside. So these are the main difference between a prokaryotic cell and a eukaryotic cell. Thank you.